Okay, so if you'll notice, we're working with a eukaryotic cell. How do I know that? It's eukaryotic, means it has a true nucleus. So if you will notice, I have a true nucleus here. Here's the nucleus. The genetic material is within that nucleus. Now, as long as the chromosomes are not in a really condensed state, they're not visible. You can't see them. And in that form, they're called chromatin, which you'll see that term right here, chromatin. That just means that the chromosomes are not condensed. They don't actually condense until the cell starts to go through cell division. And when it goes through cell division, then they have to condense. It would be necessary that they do that, or it'd be very difficult to take half of the chromosomes to one side of the cell and one half to the other side of the cell, because some of these chromosomes are very long. They're like 1 meter in length. That's pretty long. Cells are a little bitty tiny. How would you get 2 of those to go to different sides of the cell? Obviously, you couldn't. So you have to condense them very, very tightly in order to get them to go to different sides of the cell. But if the cell's not doing any cell division at all, they will be in a loose, uncoiled state, which all of them together, for us, would be 46 chromosomes, would be referred to as a chromatin. Okay, so we have a double membrane. That would be the nuclear envelope, or nuclear, which is what they put, nuclear envelope right here. And then we have, within that, you'll see that you do have pores. That's a pore. That's a pore right there. Obviously, things have to get in. Things have to get out of the nucleus. There has to be something going on there, or they wouldn't put holes in it. God would have just gone, well, no need for holes. We don't need holes here. So, wouldn't have happened. You do see a nucleolus right here. This structure here, that's where you produce ribosomes. So these little bitty dots that you see here, like this one and this one and this one and this one, those are ribosomes. We've talked about to make a protein, you have to put one amino acid to the second to the third to the fourth to the fifth and build up to at least 50 before you have a protein, which can then begin to fold and form into something like an enzyme or a transport protein. Molecule is what that is. But you have to put those amino acids in order, in specific order, like 20 next to 2 next to 7, and there are only 20 amino acids, so you put those in whatever order. You need to build brown eye color or green eye color, or a transport protein, or this enzyme or that enzyme. So most reactions have enzymes, so you got to have all these different enzymes to make that whole thing work. All these reactions happen. Yeah, you got to have all of these in very specific order. So they're placed in specific order at the ribosomes. And the DNA is what actually carries the genetic code for which order you place those amino acids in. Is it 20 next to amino acid 7, next to amino acid 12, next to whatever, or is it amino acid 2 next to amino acid 7, next to amino acid whatever? Amino acid 12 next to amino, I'm using 2s today, 12 next to 4 next to, that folds into a different pattern. So the DNA carries genes that will tell the cell basically what order to place those amino acids in. Because if you're in a liver cell, you're going to do certain things. If you're in a pancreas cell, you're going to salivary gland, you're going to make salivary amylase, which we all produce in saliva. So depending on where you are, and within the cell, you got to make a whole bunch of different enzymes so it can function to stay alive. So the DNA carries the codes, and all it does is carry the code that will tell you what acids in. That's it. That's all DNA does. It does nothing more than that. Okay, so ribosomes are very important. That is where you put the amino. Different ways you can actually put that. And then if you'll notice, we do have all these little channel systems. Now, some of these have been cut. It kind of looks like a maze of some sort. Some of these have not been cut. So if you're looking smooth endoplasmic reticulum, which would be right there, they're not actually sliced. You actually do see the tubes. And endoplasmic reticulum, that just means, endo means within, plasmic means within the cytoplasm, basically before they started using cytosol, which would be the fluid. So within the fluid of the cell, basically, is what they're really referring to. Endoplasmic, within the fluid of the cell, the cytoplasm. You have reticulum, endoplasmic reticulum. Reticulum is nothing but fibers. Because when they first invented microscopes, or really good microscopes, all they could see were little fibers in there. They didn't know that there were other fibers in. Are they showing us those? Yeah. Microtubules, microfilaments. This is part of the cytoskeleton. There are some fibers in there, but they couldn't see those at all. They couldn't see those probably until somewhere, I would think, in the 70s or maybe early 80s. I remember the first Scientific American that came out that actually had the fibers in the Scientific American. They finally developed a staining technique where they could see them. They knew those fibers were there all along, had to be, and they crisscrossed throughout this entire cell in order to keep these organelles in place, to keep everything, because gravity, of course, would pull everything to the bottom of the cell. What would keep it from doing that? You got gravitational pull, you know, everything would just fall to the bottom of the cell. You know, why aren't we a puddle of cells on the floor? Well, we got a skeleton, and then, of course, we do have our cells, which are going, the cell shape is being maintained into whatever shape it is, because you have fibers that crisscross to maintain that shape, you know. Or all of these cells would end up down here in your into your fingers and all, you know. So you, even if you had a skeleton, you know, what's to keep all this from just falling? You know, people don't think about those things. I think about those things. I've been thinking about those things since I don't know when, you know, since way back. I don't know when I started thinking about those things. Well, you know, we got gravity. You know, somebody presented the idea of gravity, and I'm going, well, you know, why am I not a puddle? And then I go, well, I got a skeleton, and I'm like, well, okay, well, I've got all these cells, so why aren't all those just, you know, way down here, you know, on this skeletal form? So I don't know. I think of weird things, and you know, but you end up getting those answers, you know, and that's the cool. Being a biologist, and some people are and some people aren't. You know, biologists are people that think of those kinds of things. You know, what's going on there? You know, and you need to find the answers to those. And the only way you're really going to do that, the easiest way to do it is to go to school, so someone can just tell you. You know, maybe it's even easier to go online now. You know, I don't know. I have no idea. I mean, you can spend from now till the rest of your life just sitting in front of a computer getting answers to things. And I even remember when they first came out with, I read somewhere where they were going to have this thing, I don't remember what they called it, they could have called it the internet, where you could just sit in front of that computer and you could just ask it questions and just get answers from now for the rest of your life, and just get any answer, just keep getting them and just keep getting them. And I thought, oh my God, I'm going to spend the rest of my life in front of whatever that is. You know, I don't, of course, but that's what I was thinking I would do. You know, after a while it gets just like, oh, I mean, you know, too many questions. Fun. You know, you've got to eat. You got to, spend all your time there. Okay. Anyway, and most of my questions in biology, I really feel like I've answered over time. It took a while to get there, but I'm there. And occasionally I run into a student who is just a natural biologist, and I can tell that they are by the questions that they ask me. I'll go, and I'll speak to them like after class and go, you're seriously a biologist, and I don't know what you thought you were going to major in, but you really need to think about biology. And there are things you can do with that, because your questions are always just the exact question a biologist would ask. And I love to meet those people. Lindsay, who's our lab tech, she's a natural biologist, just a natural for biology. Anyway, okay, back to this. I'm rambling here. So endoplasmic reticulum, which is what they first did see were little fibers, but they end up being little tubes is really what they are, and they're transport tubes. So if you want to move something around within that cell, that's where you move stuff. So the smooth endoplasmic reticulum, which is, again, right here, smooth ER, what we're doing is we're, for example, forming, making, producing fatty compounds, lipids, like phospholipids, fats, just fat we put on our bellies, or women typically put it on their hips. Most people put it on their butts, except for me. I didn't get that gene. My butt gene just like no, it didn't show up. You got the no butt gene. Like, thank you. Yeah, I wish they could suck all this out and blow it in back here. That'd be cool. Oh, down with it, Plastic surgery, sure. Bring it on. I like that. Entire. Yeah, absolutely. Anyway, okay. So not only do you make and form different kinds of lipid materials in the smooth ER, that's where you do detoxification. So to detox a compound, whether it's an antibiotic, if you take an antibiotic, then you got to take it later. It gets detoxed. I mean, they destroy it. Your body destroys it, gets rid of it. And other also as well. Alcohol, your liver, obviously. You've got smooth endoplasmic reticulum that can detox the alcohol. At the same time, it produces a chemical that will destroy cells if you're drinking too much alcohol. So be careful of that. And other compounds as well. Anyway, so now the rough endoplasmic reticulum has the ribosomes attached to it. Smooth ER does not. So these have the ribosomes attached. So that gives it a rough appearance. And the ribosomes are where we make proteins. So then it would be logical, if we make the protein out of ribosome, then we must be transporting that protein within the rough endoplasmic reticulum, which is exactly what happens there. Sometimes you can transport a protein through this rough endoplasmic reticulum, and then you can form a bud, which is really, in this case, it's a vesicle, which would be this structure right here. Ooh, I don't want that big one. This thing right here, which is just a sac. So you can put protein into that. You can do the same thing with the fats. You can, from the smooth ER, you can put fats into a vesicle right here. So this has protein in it, and this has fat in it. So I'm going to send a protein over here to the Golgi apparatus and a fat over here to the Golgi apparatus, and we're going to move those materials through the Golgi all the way through, and I'm going to package those two things together, and then I'm going to release it as this thing right here, that vesicle. Okay, so I release a vesicle, and this would be a protein and a fat together, which would be a lipid protein is really what you would call it. So I can send different things into that Golgi apparatus, named after a man, Golgi. If you actually Google him, he did a whole bunch of stuff in biology, not just locate this deal. Very important person. So And then I can use that lipoprotein. For example, a lipoprotein would be proteins. You may have heard of those. LDLs, low-density lipoproteins. HDLs are good fats, things you get in, say, avocados, olive oils. Those are good fats. And then you can eat bad fats. Bad fats, things you get in butter and animal fat. Eating a steak that has low-density lipoproteins in it. And so those are not good things to consume. But again, they do have things to help with that, like a lot of people take Lipitor. I take a combination of vitamins, which works beautifully. You guys don't worry about it at your age, and you shouldn't. You don't need to worry about that until you get to be 40 years old. And then from then on, you need to be really concerned about it. So you get your blood work done at least every 6 months, or definitely every year, to make sure your numbers are what they need to be. So you don't have to concern yourself about it, except with your parents and your grandparents, and they need to really be concerned. And if they're not concerned, then you make them concerned, in order to keep them healthy. Because, I'm sure unless you don't like them, you want them around for a while. When you lose a parent, all of a sudden it's like, whoa, that hurt bad. It's a tough one. You lose your grandparents, that's not easy. But when you start losing parents, it's like, whoa, that's really not cool at all. I'm not liking that. Don't like that. Then you start losing some of your best friends, then that's not cool either. Anyway, so you want to try to keep those that you love and care about healthy for sure. So high density lipoproteins, very good. As it turns out, women produce a 10% higher HDL, high density, than men, which is why women typically don't have as many strokes and heart attacks as men do, which is why when you go into a nursing home, you see mostly women. You very rarely see a man in there. I mean, it's not like they're not in there, but it's not that common. It's a whole bunch of women. You go, well, the man had to do the hard work, blah, blah, blah. That's not it. They play a little bit of a role, but it's mostly those lipoproteins. Men have more LDLs, women have more HDLs, just naturally manufactured. It's in the genes. It's in the gene pool. You don't ask for it. It's just the way it is. So, especially if you're male, you really need to watch that and keep your numbers where they need to be. Anyway, so that's a packaging center at the Golgi apparatus. And I don't just have to say, send a protein from the rough endoplasmic reticulum and a fat from the smooth endoplasmic reticulum. I can send a protein from the rough and glycogen, which is animal starch. It's what we store in our livers. It is starch, but they call it glycogen, and it's just sugar to sugar to sugar to sugar to sugar, just like starch is. And I can make that also at the smooth endoplasmic reticulum. And so I can send a protein and a glycogen, or not a glycogen, a sugar, into the Golgi, and what we're producing is what's called a glycoprotein. Glycoprotein would be, for example, what you form in your nose, what you blow into a Kleenex. We call it mucus, if we want to be really kind about that, or if you want to use the vernacular term, the more common term is, say, snot. So the snot. It's a glycoprotein. It's a sugar protein complex. It's actually not bad tasting. It is sugar and protein. It's very sweet. I remember as a child actually eating some of that. I know. You may find that disgusting. But, yeah, I did. I remember doing it. I can admit to that. I certainly can. I mean, sugar protein, it's right here. You don't have to, go look for cookies or anything. It's right in there. Pull it out, slam it down. There you go. Got my sugar. I don't think I made a real habit of that, but yeah, I've tasted that for sure. So anyway. And I think I'm not the only human being to have done that, but I may have been. Yeah, and I don't make a practice of that anymore. But that's a glycoprotein that we do manufacture. So it's manufactured at the Golgi apparatus. It's a packaging center. You put a couple of things together, sugar protein, lipid protein, maybe a sugar and a fat. That you can do that as well. So many different things you can package. And then you can form a little vesicle like this one. We formed a vesicle here. And then you may want to transport that vesicle out of the cell. For example, the snot, our mucus that we produce in the nose. Maybe we make it in a cell lining the nose, and then we want to transport that particular molecule out so that it is now within the nasal cavity. And now it's in your nose, so you can blow it out into a hanky. Or most of us just do that, which means I just pull snot down the very back of my oral cavity, which it went down my esophagus and landed in my stomach. You know, that drops that particular glycoprotein, which is snot. You eat it all day, every day. Anyway, you just don't pull it out here and put it in here. You're eating it. It's going down your esophagus into your stomach. You eat snot every day, all day. So as a social practice, we say it's not socially correct to pull it out here and put it in here. What's the difference? Nothing. You're eating it anyway. Yeah. I'm not condoning that you teach your children and that you yourself, eat snot, but I'm just saying you do. Yeah. Anyway, somebody had a question. Sure. Why does it make you nauseous? That may not be what's making you nauseous, the glycoprotein there. I wouldn't think that would. It's just food is what it is. I wouldn't think that would. I mean, it is food, basically. Your body digests it and absorbs it, and you use it as food. Not a bad thing. But yeah, you make an abundance of it for sure when you're sick, typically, depending on what you're sick from. But yeah, you export it. It goes out in the nose. Goes out into the nasal cavity. Okay, let's see what else we've got in here. This is, again, an animal cell. We did the nucleus. We did the rough ER. We did the smooth endoplasmic reticulum. That's got to be somewhere. Are they not giving it to us? Yeah, here it is over here. Smooth endoplasmic reticulum. They put way over there. I'll go back to red. That looks a little better. These structures that you see right here, these are, because I have one here, one here, so I only have them. Those are called the mitochondria. One is a mitochondrion. If I have three, it's mitochondria. So leave off the I-O-N and just put a mitochondria. That's plural. This means you have two or more, 2+ or more of those. Those are where we produce the ATP. So each one of these is actually cranking out a huge amount of energy, ATP energy, and again, like I've said, that's what we use as an energy currency. Anything you want to do, if you want to do a backflip off the high diving board, or just a backflip off the side of a pool, or dive into the pool, or swim through the pool, or float on a noodle with a cocktail in your hand and just kind of float around and drink the cocktail. Just the movement of moving the cocktail from your hand into your mouth. That takes some ATP. It is some exercise. That's, you're exercising there, as I see it. Just so you'll know. All right, but anyway, your ATP. And interestingly enough, the mitochondria, you only get those from your mom. Your mom carries those in the egg. The only thing that enters that egg at a fertilization is the head region of a sperm. Now, the sperm looks like this, and it's got this what they call a neck region, and it's got this long flagellum for motility. But the mitochondria are in this region right here. So it gives it the energy in order to swim. The head region, which is the region right here, that carries the DNA, that carries the chromosomes, carries the genetic material. The only thing that actually gets into the egg is this section right here. That, just the head section. So mitochondria from the sperm into the egg, so the only mitochondria that you're getting are ones that were in the egg. So the mitochondria associated with sperm are down here. Those don't get in at all. So all of your mitochondria came from your mother, which is interesting. Which hers, those came from, and so on. Mitochondria. Might as well look at it like that because I don't know what it is. Which I think, you know, if you look at it like that, that's pretty cool. Okay. Other structures. Let's see. I'm about out of color, so I'll erase all that out of color. So let's look at the lysosome. The lysosome is actually manufactured apparatus. It contains digestive enzymes. So if we're bringing something into the cell, now this is probably, it could be really moving something in to the, so maybe I have something here that's, you know, I don't know, that we're trying to bring into the cell. You know, so once we bring it into the cell, then it's this thing, and then that will fuse with the lysosome, and then it will digest whatever this particle is. Your white blood cells engulf bacteria. They can engulf viruses. If you happen to introduce pollen or mold spores or anything foreign, your Golgi will produce lysosomes within a white blood cell, and the white blood cell will engulf whatever that foreign agent is, and then the lysosome will fuse with that foreign agent, bacteria, virus, pollen, dust, spores, whatever it is, and destroy it. You don't want things that don't belong in your body to be in your body. Obviously, we don't want that stuff there. So we need to get rid of it. This is just the way to do it. Lysosomes. If you've ever tried to catch a lizard in West Texas, I don't know if you've ever done this. Certainly I have on many occasions. You see a lizard, so you try to be real still and just grab it by its tail. The tail will drop off, and it's just wiggling, you know, like this, and you think, oh man, you know, if you're an animal who's not as bright as we are, you know, an animal's like my cat will go, cool, man, this is great, look at that thing, you know, and the lizard runs away. Well, the lizard will make another tail. The cat's like, well, that's hardly worth eating. You know, by that time the lizard's gone, you know. But what happens, you know, when you grab the tail, you know, he knows that. So from his brain, he sends neural impulses to a section of the tail, and all of those cells automatically release lysosomes. So you destroy all the cells within a plane. So all those cells are just totally destroyed, and then the tail drops off. Kind of cool. You know, and then that lizard survives. So that's a survival mechanism, very cool. If you have something that wears out, say like a mitochondrion, they don't last forever. If they wear out, you need to get rid of them. So a lysosome destroy a worn out mitochondrion or anything within that cell that is not value, that isn't needed. Why keep it? What's the point? Just get rid of it. Okay. What are these things? These are called the organelles. We're looking at the organelles, the ribosomes, the Golgi, the smooth and rough ER, so forth. I already said ribosome. Anyway, even the nucleus is considered in most books now to be an organelle. They didn't for many, many years consider it to be an organelle, but now it is an organelle. Okay, check my belly button. No, joking. All right. So, green light. Green light. Okay, so what else do we have here? We talked about lysosome. We have the peroxisome. That thing will actually break down hydrogen peroxide, which is something cells do not need to have in them, and cells produce them. Bless you, bless you. Okay, just waiting on the next. And so we make something that can surround that hydrogen peroxide, and then we can really use a lysosome to diffuse it at the peroxisome. So we can destroy that also. Okay, other than that, we do have the centrosome, which really the centrosome is the entire area where the centrioles. The centrioles, you'll notice, are kind of barrel-shaped. These are really long tubes. So, and they're at right angles. So you have tubes that run this way and then tubes that say run that direction, basically, trying to make right angles of tubes. And they do play some role in animal cell division, but plant cells do not even have them. So they're not. They're manufactured within an area called the centrosome. So the centrosome is where that would be assembled. And good enough. They're like 100 micrometers in length, so they're fairly long, typically. No, that would be, I'm thinking of something completely different. I'm not coming up with it. I'm trying to think of the diameter of a microtubule, and it's not popping in. So I'm not going to worry about it. We'll get to it eventually. So microtubules are like soda straws. The microfilaments are not at all. They are very, very tiny. They're very, very thin, obviously. Micro means very small, and it's just a filament instead of a larger tube. And all of these form kind of microfilaments basically form like this mesh-like work beneath the cell membrane. And what that does is maintain the shape of the cell, help maintain the shape of the cell. And then, so all of these are microfilaments here. Microtubules will form things like, as an example, this thing, the centrioles, those are microtubules, and some of the things that we'll get into at some point also will have microtubules in them, but we're not getting into those. Intermediate filaments, I don't usually discuss that at all. So if you're interested in those, it's in the book. I'm not too terribly interested in that, so I'm not going to worry about them. If you're concerned, you're welcome to spend your time worrying about those. I'm not. Okay. So, and then the last thing, of course, is your outer boundary to the cell, which is the plasma membrane. That's your plasma membrane here, and it's how things get out through that cell, come in and leave at that boundary, and so something has to do that. We certainly don't want anything to be able to get in. We certainly don't want just anything to be able to leave. So it's sometimes called selectively permeable. differentially permeable. I've heard it placed given that definition also, but selectively permeable works really well. All right, so let's go to the plant. It will have everything the animal cell has, except the animal cell has the centrioles. Plant cells do not have centrioles, although the cells do divide, obviously. It has everything else that we discussed. And then it has three things that the animal cell does not have, and those three things would include this big, huge sac. Sac is the vacuole. So in the central vacuole, that's where we store mostly water. You know, if you've had plenty of water, then all the grasses will stand upright. But if you don't water, all the grasses and the weeds and so forth, they'll just kind of begin to wilt over. So what happens is, if you got plenty of water, you know, you got a lot of fluid in here, so it's creating pressure, you know, up against all of this. So, you know, it's a very filled cell. But if you don't water the plant, then the plant will take water out of the central vacuole because it uses it in the chloroplast to make sugar. You got to have water to make sugar. All right. So if you don't water your plant, it'll get it out of this sac. Well, when you drain this sac, of course, it's gonna shrivel up. That will make the whole cell kind of shrivel up, and that's just one cell. So if you got the whole plant, you've got millions of cells, and if they all pull the water out of that central vacuole, all of the cells shrivel up. So of course, the plant's going to wilt. I don't know if you've ever thought, why does the plant wilt? What's that about? Well, it's wilting because it's running out of water. And if it completely runs out of water, it's probably going to die because it's got to have water to produce sugar. Sugar is its food. If you don't get food, you do not make it. Ladies, please, if you want to talk, have your fun out there. If you want to strip off naked and run around, go ahead, get it over with. I guess not. Okay, I gave you your opportunity. I was just making fun, obviously. But you know, and seriously, I'm okay if you all want to sit and chat, and we can chat for as long as you want. I'm all for it. I would love that, absolutely, because I like to have fun more than anybody and chit-chat. Believe me, I'm the biggest chit-chatter you've ever met. I love it. But in here, I can't allow you to do it because of these other people. I don't mind it. I've already passed a class. I've already got 3 diplomas. You know, it's all cool for me. Whether you're disturbing the class or not, my paycheck will continue to roll in. This has no impact on me, 0 impact on me. It impacts other people who are in here, and that's the point. That is the point. Just keep that in mind. I'm not trying to be an ass. I'm not trying to be your father, and you know, nothing to do with it. I'm just trying to protect. I don't use, when it comes to, if you'll notice in the lab, they said the animal cells have small vacuoles in the lab book. Well, that's Sylvia Mader. Mader, just poor girl. Poor Sylvia. Should we just all bow our heads for a moment? Talk to you for so long. Conversation, please. Anyway, but in animal cells, I don't use small vacuoles. I just use vesicles. But yeah, no, you do have a central vacuole in plants. You know, and it can store other things. There are other things you can put in there. One thing that you will never ever see in a biology book, or maybe you will, maybe they'll put it in, but I've done enough looking around that I have found that in a central vacuole, that's where they put their wastes. It's like a waste deposit. So they don't want waste out in the cell. And in an animal cell, you can transport all the waste out. So it enters your blood goes through your kidneys filters all that, and then it goes into your urinary bladder, and then you get rid of it. You void it. Okay, so, you know, eventually we can get all the waste out, and that's cool. But in the plant cell, you know, you've got this big, thick, huge central vacuole here. I mean, big, thick, huge cell wall here, and you can't just get rid of stuff. So, you know, waste materials are stored in there, which is kind of cool. A place to just dump all that stuff. And I think that's interesting. Makes sense. Okay, then the chloroplasts, obviously, we don't have chloroplasts. That would be great, you know, if we were green. People talk about little green men, you know, from Mars or whatever. I think if we could put chloroplasts in our cells, and, you know, every time the cell would divide, it would make more chloroplasts, and so we'd always have chloroplasts. It would be great. Put the gene to make chloroplasts somewhere in our DNA, and then we would never have to buy groceries again. You know, it came time for lunch, you know, you just go out in the big, huge lawn out there and strip off and get naked and get some sun, you know, and lay there for an hour and just pull in the sunshine, mix your sugar, get filled up, come back to class. You know, I think more people would stay on campus at lunch. Make lunch a little more interesting, for sure. Okay, anyway, sugar is produced. That's where photosynthesis occurs. Plants take in carbon dioxide, usually from the air, CO2. And, of course, they have to have water. Wherever they're going to get that, hopefully from the rain or somebody with a hose, hopefully it's from the rain. And if they don't get it from any of that, they're going to pull it out of that central vacuole. But they use all of that and put it together, and they make sugar from that. C6H12O6, C6H12. Bad. Anyway, they make sugar. And they release oxygen. Plus, they make some oxygen, which is great. You know, most of the oxygen is in excess. They make way more than they need. But they do need oxygen, just like you and I need oxygen. We have to breathe to get our oxygen. The cool thing about plants is they make their own. They don't have to take in oxygen. Not necessary. Not only do they make their own sugar, they make their own oxygen. Cool. Very sweet deal. Very nice. Very, very cool operation. A plant. And, you know, most people are not all that interested in plants, and I just love them. I'm like, what? You have the ability like to just grow and make your own food and make your own oxygen just out of things that you're getting out of the ground and the air? Really? And just grow and be a big old tall tree, you know, a sequoia redwood tree in Northern California that, you know, you could park a yellowhound, the school bus, on a branch of one of those trees. They're huge. You stand under one of those and you go, God is good. You know, you don't have any other response. It's like, whoa, too amazing. Anyway, so yeah, and pretty amazing. Okay, the cell wall, obviously that helps maintain the cell shape, but it does kind of create a barrier there so things can't get in and out. Although sometimes you can see you've got holes. This isn't always. If they have holes, these holes are called plasmodesmata. Not all of them, so I put a plus or minus, meaning some plant cells will have those and some won't. So things can move back and forth between cells is what can happen with a plasmodesmata. We have some cells that do that too. Our heart cells have little passageways for things to move back and forth, which gives it its function, allows it to function the way it does. Holes, it wouldn't function the way it does. Okay, so that's your plant cell. And let's go on and see where we are. We are at no, in fact, plasmodesmata, in reality, I had never even seen that on the slide. Never paid any, I didn't even know it was there. But I started talking about that, and then I looked up, and it was right there. It's like meant for me to even talk about it. This, for some reason, it was like, bam, there it is, right in front of me. Just eye level, plasmodesmata. I'm like, whoa. Cell wall. So you got a central vacuole. You have chloroplast, and you have the cell wall. Central vacuole, chloroplast, cell wall. You have three things that you find in a plant cell that you do not find in an animal cell. Okay, I know, we're out of time. I always run over. I know, I'm a bad person. I'm sorry. You guys have a wonderful day. Enjoy. Life is short, so enjoy all you can. Laugh a lot.